machine learning coffee session. So this week we will have a talk by Saed khawaldeh from the University of Oxford, and he's going to talk about brain structure activity dynamics. If you have any questions, please go ahead and just write them in the chat, and then I will then forward them to speaker. So Saed, go ahead.
Hi everyone. First, many thanks actually for the invitation to give a talk at the AI Institute, and it's actually an honor to be here between you guys. So just as Marcus mentioned, I'm actually in the last year of, in the last few months of my PhD at the University of Oxford, where I'm actually working mainly on utilizing machine learning techniques to analyze brain signals, specifically LFP and EEG signals. So in today's talk, I'm going to be actually talking mainly about the piece of work which got published recently this year, in February, at a Brain Journal, and it is actually about using the brain structure, activity dynamics, to do limb movement, a prediction, for the Parkinson disease patients. So just to start, just saying, so mainly the problem which we are trying to address by this work here is actually having paralysis in general, or having limb loss. And as you know, there must be some devices to help people who have some sort of physical disabilities to live in a normal life, and just try to overcome the difficulties which they have. So according to some stats, actually, in this paper, they're actually mentioning that there is 1.7% of the US population, they have actually sort of paralysis or physical disability. And actually, annually, there are about 300 billion US dollars are spent on that, in terms of treatment, and also having a nice places which are supporting the people who has some physical disabilities. Just jumping, obviously, thinking that we have physical disabilities, so how we can solve it. There are the traditional systems which they are using muscle activities, which is EMG, and then basically try to predict the actual movement what the patient wants to do using the muscle activities, the EMG. But there are actually the next step, which is a bit we heard about in many of these cool, I would say, talks from Elon Musk, where it talks about using the actual source of signal in the brain, and try to help patients who have some physical disabilities. And here, when we talk about using brain signals to control or help people with physical disabilities, we're talking about something which we call brain-machine interface or brain-computer interface, BMI or BCI. And here, I just want to mention that obviously, we have the human brain, which has billions of neurons, and those neurons are talking to each other using some sort of electrical activity, which we can capture using different imaging methods. So there are some imaging methods like the ECOG, where you need to install a grid of electrodes or sensors on the surface of the brain, and you need a surgery to do so. And you could actually use an implant, which is similar to what the guys in Neuralink are using, or you could actually use EEG signal, which is basically a signal calculating the electrical activity which are generated from the population of cells near to that electrode. Or there are the things which are a bit more kind of invasive, which is using implant LFPs, which is implanting a sensor deep in the brain structure, going to the specific brain structure which is relevant to certain tasks of the human body, then trying to record from that specific brain structure. And in our case here, we're actually using the subthalamic nucleus brain structure, which is a deep brain structure in the basal ganglia, to do recording from small population of cells up to 50, actually something about 50 neurons, where we have sensors recording from that specific brain signals or brain cells. And once we have recorded these brain cells, we should do some sort of pre-processing to trim the artifacts, the noise, either it is electronic noise, which are because of the devices which you're using, or it is electrophysiological noise, which is because of maybe heartbeat or physiological features which we have like because of the human body or movement, maybe like artifacts. So once we have done this like pre-processing, we can extract features, then use a certain decoder or like a classifier to use these features which we extracted from these LFB signals from the brain to control a wheelchair, or a computer, or a robot arm. That's basically the general structure and pipeline of the BCI and BMI systems. Just talking about the study which I've been working on like in the past year, and has been published recently. So actually we're here like going deep in the brain, like to a brain structure which we call which like it's called subthalamic nucleus, and there is this electrode which is implanted there. The electrode looks something like that, which has like 8 sensors, as you can see. We call this one directional electrode, because as you can see, like we have these different like electrodes on the sides, which allows us to do like recording of the cells which are closer to that one. I mean, less of the cells which are a bit far from that specific electrode. So that's mainly like the experiment setup in terms of like in terms of like what device we're using for the recording. I would like to mention that all these patients which we have recorded, the 18 patients, they're actually like PD patients, Parkinson patients, and we're allowed to do like such recording only because they already go through the DBS, deep brain stimulation like surgery. And DBS, for people who doesn't know what's DBS, it is just a surgery where they implant this electrode inside the subthalamic nucleus for people who has Parkinson, and they use like some electric activity or electric signals which they send to this subthalamic nucleus in different amplitudes and different frequencies, trying to suppress the Parkinson symptoms. So some Parkinson patients, they might have mainly like rigidity with their movement, and when they send like some electric shocks or electric signals to the subthalamic nucleus with certain like activity and certain frequency, after they try obviously like various like combination, when they do the programming of this electrode, they actually can suppress the activity, like the symptom which are related to the Parkinson disease. However, in our case here, like we're not actually working on like sending electric shocks to the brain. We're actually recording from the same electrodes which they are like which they're supposed to use to send electric signals to. But here, like we're doing this recording right after the surgery is done. There are a window of 3 days where the patient is waiting basically like the programming of this electrode. Then we utilize this time and we do the experiments which we want actually to do, like here, for example, assessing the difference between upper and lower limb movement, and can we really predict movement using like brain signals or not. Like the experiment paradigm which I want to actually like show to you today. So once the patient had that surgery, and then the patient is externalized, where we have the signal from their subthalamic nucleus, we actually ask the patient in first paradigm, which we call fixed block paradigm, to do like a series of certain like type of movement for 20 to 30 times. As you can see, like upper limb movement, which is just a hand like movement, or like a feet movement, as you can see here, which is like a lower limb movement. So they do the patient do that like just in a block of movement. So here, like they get a go cue, which is like a acoustic cue. Once they hear, they start doing the movement, which they know that they're gonna do upper or lower limb movement. So that's the fixed block paradigm. And the second paradigm is we call the randomized block paradigm. And for this one, we actually ask the patients again to do like upper and lower limb movement, but instead of doing that in blocks, we actually did that trial by trial. So instead of giving the signal go only, we're giving them the signal hand, like maybe leg. And then the patients were doing that randomly, like we actually give them a random signal. The reason we're doing that, because in the previous paradigm, the patient know for the whole block of movement that they're gonna do like hand movement or like leg movement. But here the patient doesn't know before, like the acoustic cue. They know that they're gonna move, but they're not sure, is it like a hand or like a leg movement? And that's what they know in the acoustic cue, like here. And then we record obviously the EMG and the accelerometer and the LFB signals, which are from the subthalamic nucleus. The EMG and accelerometer signals, which are actually the muscle activity, is for us to have like a kind of grounded truth, which allow us to do the labeling later on. So once we have actually got that, we actually like collected the signal, we processed it by doing like some band pass filtering. We were interested, I actually know, we did a high pass filtering, and then we downsampled the signal and did the similar, like the small artifact removal to ensure that the signal is clean. Then like apart from like the main objective of this study is actually, as I mentioned, like to do upper versus lower limb classification. Can we really predict movement from the LFP signal, or we can't? That's the first like objective which we wanted really to achieve. The second one, in the Parkinson specifically, it's reported in the literature, there is a hyperactivity of something which we call beta bursting. And beta bursting, for people who don't know that, you can think about it as like, you know, the heartbeat, which is like simply like a peak of the heart signal, ECG. So there are similar things which happens at the beta frequency in the brain, and these like are proven in literature to be associated to like to the pathology of the Parkinson. So Parkinson patients in general, they have extra hyperactivity of the beta bursting. That's why like researchers and in previous like literature, they think that's why they have like some symptoms like a tremor or like rigidity or slowness of movement. So the second objective, as I mentioned earlier, we actually wanted to study what is really the effect of these beta bursts on the classification of movement, where the classification of movement, the accuracy of the classification of movement is dealt with as an indication of the movement impairment. So for example, if we could do a good classification of movement, this means we have less like movement impairment. And if we could do poor classification of movement, this means we have a higher like movement impairment. So once we have done this kind of burst versus non-burst detection, and we use for this like this method where we're doing simply like 75 percentile thresholding. So as we extract the alpha envelope and then we do 75 percentile thresholding, then we get like the both like the alpha and we got the alpha like, you know, bursts, which are these like periods where there is elevation of alpha activity. Or we do the same thing with the beta, and then we detect the beta bursts who are like longer than 100 milliseconds. And at the end, when we detect all these bursts, to basically for the sake of like doing the second like objective of the study, which is assessing the effect of the beta presence on the accuracy, we have actually like done this by specifying the beta periods, the alpha periods, and non-burst periods. And here non-burst, I'm referring to like the periods where there is no alpha nor beta. It's like a flat signal somehow, as you can see here. So that's like regarding the detection of bursts. Then like after that, either like in the first paradigm, in the first like objective, when we wanted to classify upper versus lower limb movement, or actually when we wanted to assess the burst effect, we did feature extractions. As you know, it's a straightforward, like if you want to do any classification, you need to do feature extraction. And here we have extracted the power activity of the frequency bands between 1 and 500 hertz. And we've extracted also other statistical features like the mean, standard deviation, and these different things. And these all the features which we extracted for every single trial. And then we use naive Bayes algorithm to do classification of limb, like limb, like for the two conditions, like an upper limb movement or lower limb movement. Just moving to the results, actually, that maybe talking about the first piece of results, which is related to the classification of upper versus lower limb movement. Just first, I want to mention that, as you can see in the time window here, we have like 4 different task periods. Resting, when the patient doesn't do any movement and just waits. Pre-Q period, which is basically when the patient got to know that now they're going to make certain movement. In the fixed block paradigm, which I mentioned earlier, they know that they're going to make an upper limb movement or lower limb movement. Then we have like the minus onset, which is after the patient got the go cue, but before the actual execution of movement. And the plus onset, it's like the period when after the patient has started doing the execution of the movement, either like a hand movement or like a leg movement. And what you can see here, every single dot represent one contact. Here we show the directional contacts, which are the sensors, and we have the mean in black, as you can see. So for the fixed block paradigm, as you can see, in the rest like period, we're getting something around like 50%, which is random classification of upper versus lower limb movement. However, once we go to the task block, when the patient know that they're gonna do upper versus lower limb movement, we get something around like 80% or 80+% area under the curve for the classification of upper versus lower limb movement. I want to grab the attention here like for like the minus Q period. The minus Q period here, the patient know that they're gonna make certain type of movement, like upper or lower limb movement, but there is no actual execution of movement. So they haven't done the movement. So here, like, it is like an encoding of, I would say, like intention of movement for this period. And this is somehow really interesting that we're not, I mean, it is like obvious that we can like classify the movement when like the patient is doing certain type of movement and moving their arm. But actually the thing which is really interesting is doing the classification of the movement during the thinking of the movement, when the patient is intentioning to move like either like an upper limb or lower limb. And this is the contralateral results for one subject, and this is the ipsilateral results. So this is again for the fixed block, when the patient is doing like group of same movement like after each other. Moving to the second paradigm, which is the randomized block movement. And for the randomized one, as I mentioned earlier, the patient doesn't know about the actual movement which he or she wants to do unless they hear the go cue, because the go cue tells them upper limb or lower limb. And the go cue, it's actually like here at like the minus onset. So as you can see, the thing which is really interesting from this one subject results, we get something really like close to random, like in the PQ period. And here, like you can see, it's like something which is really like close to 50%, which is random, like in the pre-Q period. However, it goes up and we can actually get something above 80% area under the curve after the patient get to know that they're gonna move upper or lower limb movement. So this is the average across all the subjects which we have. And you can see in red the fixed paradigm, and in blue the randomized paradigm. And for the fixed paradigm, as you can see, we're getting like an 80% accuracy at all task periods, even before the actual execution of movement. However, like for the random like paradigm in blue, we are actually getting only the 80% or like something like 75% only when the patient is know about which type of movement the patient will be doing. To take back home, I mean, this is the kind of message which we want like to highlight here, that the patient like knowledge or intention of movement neuromodulates the STN, which is the deep brain structure which we're talking about, in a way that it can like preserve information about the actual movement which the patient is intending to do after maybe like a few seconds. Here the prediction is actually up to 4 seconds before the actual execution of movement. The second like piece of results, or the second objective which we wanted actually to look at is what is the effect of the presence of beta bursting, which are like pathological bursting, on the movement prediction accuracy, which we perceive it as like an indication of movement impairment. And as you can see from the results here, so we have the three like periods, which is no burst, alpha, and beta. And what we're getting here, we're getting that when there is the beta burst, we're getting the lowest like possible accuracy, as you can see, something around 60%, which means that beta burst presence affect negatively the prediction of movement, which means there is a higher impairment a higher motor impairment when there is a beta activity. However, like when there is no burst, we have a much higher, I mean significantly higher accuracy, like area under the curve. And we actually have looked at alpha specifically just to contrast that and just to have like for a sanity check. And what we found out that this alpha specifically, I mean, it is distorting the movement. I mean, it is like somehow it has a negative effect on the actual movement prediction, but it's not as bad as like the presence of the beta activity, as you can see from the results here. So just to conclude, like for this like piece of like work, we actually like the low frequency like 8 to 30 hertz like burst activity, they're actually like impair the LFB like base prediction, and and we can see that through like the low area under the curve, which we can see like in the accuracy. And for the beta burst, like which we found it does impair the prediction more than the alpha burst. And the directional electrode specifically appeared to give really like a very like good amount of information, which allows us to do better prediction in the absence of movement, which is like the intention of the movement in the fixed paradigm. And the combination of like these like directional electrodes and like the feature extracted from them with the basic machine learning technique which we have used, which is here like naive Bayes algorithm, are actually like they might like be used hopefully in the future for like a neuroprosthetic application, because the results shows that there is a possibility that we can do a full prediction of movement, like I mean even if it's like a upper versus lower, like using only LFP signal without using any EMG. I just would like to mention here, like regarding the algorithm which you have used, I've actually tried like probably like all the traditional machine learning algorithms like SVM, like KNN, like all the logistic regression, naive Bayes, like all these, and even decision tree. And the results I was getting all are similar to each other, but we stick at the end with the naive Bayes because like we have here limited number of trials, given that this is really like an experiment, like, you know, when the patient is going through surgery. And what we did, I mean, we just like used this like naive Bayes algorithm, which is basically like relying on the conditional probability to do the decision, like regarding which, like, or the prediction regarding which like as upper or lower limb movement is happening. So that's like more or less about everything I wanted to share like regarding this study. Just to thank my supervisors and my group members and our collaborators like in UCL and in like Mainz and in Bern on their like help with the, and Berlin also actually, on their help like with the data, and just obviously like all the patients who are actually like participating in our studies. And I'm actually like available like now for any question if anyone have any question.
Okay, thank you very much, Saed, for the very interesting talk. Are there any questions? Okay, so while we wait, I have a question. So I was wondering, so in the first study, I think you mentioned that the naive Bayes turned out to be sort of roughly as good as other machine learning classifiers. So why do you think that there was no significant differences there?
I would say like I remember I've run like about 8 like different machine learning techniques like KNN, like CVM, like the results all are like similar to each other. So why they're not like different from each other? I would say like, you know, I mean it's a kind of like a good sanity check to try different machine learning techniques and get the same results, just because you're guaranteeing. I mean, obviously I've done like training and testing and it's like cross-validated the results. They're not like in like basically like the training like session, but just like given that there is no really significant difference between like the different machine learning techniques, it seems that, I mean, this is the best which we can do with the current data which we have. So there is no, I mean, every single one of them have done like good exploitation of the features which we have extracted. I mean, I would like to mention actually I've tried neural network with that, but actually it did bad job because the data were not enough. I mean, I would assume like to do like the training for the neural network, because like the back propagation and these like kind of complex, it needs like a much bigger like amount of data in order like to train it and to have it like, you know, ready like for doing the classification and the testing like subset. Okay, thanks. So Lu Cheng has a couple of questions. So he asked that what are the alpha and beta bursts? And he also asks, why do we want to see them separately? Sorry, can you say it again? So what are the alpha and beta bursts, and why do we want to see them separately? Okay, yeah. So as I mentioned earlier, specifically the beta bursting, from the literature, that many studies are actually talking about the association of the beta bursting with the pathology of many diseases, including Parkinson. So that's why, like, I mean, given that it is associated, I mean, if patient has Parkinson, you expect that patient will have hyper beta activity comparing to a healthy patient, to a healthy person. So that's why we're looking at beta like bursting specifically, because it is like a hyperactivity which we have in the brain. like at certain like a frequency like band, which is the beta bursting. So that's why we want to look at like beta bursting. So alpha actually specifically, there are many studies which are actually talking about alpha, and it is associated with, I guess, sleep studies. And actually we've used alpha specifically because we wanted to contrast the beta like bursting effect with the alpha, just like to contrast that with each other. And I would say like the results, they were very similar there, because the thresholding method which I have used, it doesn't like guarantee like not having leakage from certain frequency band into another. So more or less like there must be some leakage from the beta like frequency band into the alpha, and that's why we're getting somehow similar effect, even that beta is worst. I hope I answered the question.
Yeah, okay. Any other questions? I think if not, then okay, we still have one question from Said. Would you like to ask this yourself, or should I read it?
Yeah, I can. I mean, I can just stop sharing screen and see it. Yeah,
So
for sure.
maybe I can read it. So okay, so Said is asking how much knowledge of neuroscience someone needs with an engineering background to enter this field.
To be honest, I'm from engineering background, and I've done a lot of work with deep learning, like and like different machine learning techniques. I mean, I have a few papers published in that field, and I was ignorant about like neuroscience when I started like the PhD. But to be honest, like it's a bit of like reading mainly, to just like do a little bit of reading, like in the area which you are specifically working on, to understand the different like I remember I've read a lot about the subthalamic nucleus and basal ganglia, just to understand how these different components inside the basal ganglia are interacting with each other. So it's mainly like gonna be like just a little bit of reading. I mean, mainly like probably papers would be the best thing, and the easiest and fastest.
Okay. Okay, so I then would like to thank the speaker again, and also thank the audience for their interest.